What we're going to do today is continue on with a particular type of randomization and permutation tests that's particularly useful for situations where the data is complex or where there are some unanticipated challenges in the data. When we talk about randomization tests, what we do is take advantage of the fact that we had an experiment, that we randomized which units are assigned to active treatment and which units are assigned to control in order to get a reference distribution for the test statistic. In other words, to figure out how surprising our data would be if the null were true. And then a permutation test is just applying the algorithm for a randomization test to a situation where you actually did not randomize. But here's the situation we're going to talk about today. And we'll imagine we have a very small study of only 4 people, but of course this could be generalized to a larger study. Suppose we're comparing the effectiveness of an old drug to a new drug. We're trying to get our new drug approved by the FDA, perhaps. And suppose we have 4 people in our study and we put 4 names in a hat and we drew 2 to assign 2 to the new drug and 2 to the old drug. So we're using complete randomization here. And suppose we measure something like how many months the people lived. Okay, how many months did they live after we assigned them to new drug or old drug? And you can imagine this is a drug for some disease that is particularly progressed. So these people are not going to live very long. So suppose that the 2 people who were assigned to receive the old drug lived for 3 and 7 months. One of them lived for 3 months, one lived for 7 months. And suppose the 2 people assigned to the new drug had 0 and 12. So one of them did not live for even 1 month, the other one lived for 12 months. Given these values, we could conduct a randomization test just as we did earlier. We could write out the numbers. We could notate the treatments A and B. We could note that what we have here is A, B, B, right? We could calculate the difference in the mean number of months for A and the mean number of months for B, and then do the same thing, for example, for other possible ways to allocate 2 people to treatment A and 2 people to treatment B. And we know how to do this. We can go through and get a difference in means for each possible random allocation. We can make a histogram of those differences in means, and we can see where the difference in means that we actually observed would fall on that histogram. In other words, we could compare our test statistic to the reference distribution to get a p-value, to see how surprising our data is, given that the null is true. But now let me complicate this just a little bit. Suppose that our data collection ended 1 year after we assigned treatment. In other words, we're not going to have the resources to continue following all the people in our study until every single one of them dies. Maybe we cured one of them. Maybe one of them got better. So in particular, suppose this person right here didn't necessarily live for exactly 12 months, but rather this person lived for at least 12 months. When the study ended, the person was still alive. So all we know about this person's outcome, the number of months they live, is that it's bigger than 12. What are we going to do? Let's think about that for a moment.